Hello and welcome to a special edition of Meet the Microbiologist. Our guest today is Dr. Laura Hug, who is the winner of CSM Thermo Fisher Award for 2022. This award recognizes the outstanding contributions of early career researchers to the field of microbiological sciences. The award is sponsored by Thermo Fisher Scientific, and we thank them for their continued support to the Canadian Society of Microbiologists. Dr. Hug is an assistant professor and Canada Research Chair in the Department of Biology at University of Waterloo. Dr. Hug uses a combination of molecular biology, metagenomics, phylogenetics, bioinformatics tools to study microbial diversity in environments, particularly those impacted by human activities. Dr. Hug is considered an international leader in the study of uncultivated microbial world. Welcome, Dr. Hug, and congratulations on your award.
Thank you. I'm very excited to be able to receive this award at our next conference, and I'm excited to be here today.
Great. So to start off, Dr. Hug, I'm just curious, what got you interested in microbiology, and how did you end up where you are today?
It's an interesting arc, actually, because it wasn't very linear. I started my undergrad at the University of Guelph in marine biology. I'd always liked biology, I'd always liked science in general, and I liked that the Guelph program was a general first year, so I could take some chemistry, take some physics, take some biology, and start making decisions at that point. But I did start in marine biology, and then I changed my major about 5 times, finally landing in genetics. I really fell in love with the idea of genes controlling function, Punnett squares, your sort of basic genetic tenets, and then evolution in general really caught my heart. So I moved and did a master's in deep eukaryotic evolution, looking at protists and the eukaryotic tree of life, and really trying to date some of those big radiations and figure out how they interact with each other on a phylogenetic tree. And at that point, I still wouldn't have considered myself a microbiologist, although at this point I was finally starting to work with some microbes, although larger scale ones. I had a protist called, that was a Jacobid. And then I decided that I loved that toolkit. I was happy to have done that work, but I wanted to do something with a more of an applied focus. And so I moved and did a PhD with Elizabeth Edwards at the University of Toronto in bioremediation. And that was really the first time that I worked with bacteria outside of a second year microbiology lab plating E. coli or transforming some things in a genetics lab. And slowly started to build up my knowledge of the bacterial side of the tree of life, the archaeal side, understand microbial metabolism. I had avoided all of the microbial metabolism and bacteriology courses at Guelph. I didn't think they were interesting. And I think it would have been really helpful for where I am now to have actually had some of that taught to me, as opposed to having to kind of learn it through the arc of my grad school and my postdoc. So now I consider myself primarily a microbiologist and primarily interested in novel microbial metabolisms and interactions between different microorganisms within a community at a contaminated site, with still some interest in expanding our understanding of microbial diversity and evolution. So a lot of that early training influences the questions that I ask, but my time in an engineering lab has kind of pushed me back towards that more applicable side. So it's a bit of a mix, but I'd say it's really only the last 10 years that I would have considered myself a microbiologist.
Wow, that's really interesting story, and actually, really nice to sort of hear for some of our younger students to see how our career can take shape as long as you're open to ideas and open to trying different things.
Mm-hmm. Well, and I use all of the different tools that I picked up along the way. It's just the questions that I ask have shifted over time. And I think that's a really exciting component of a career in science.
Yeah. No, that's really interesting. So your research in your lab, super interesting research. I mean, one of my favorite papers, all-time favorite papers actually, is one of your papers, that basically just redesigning the tree of life. And I use it in my teaching, and I'm sure a number of others do as well. So tell us a little bit about what's going on in your laboratory.
Yeah, so my research group is largely looking at microbial communities at contaminated sites. We have a big focus on municipal landfills. I think landfills are a really interesting, heterogeneously contaminated site. They're engineered for about 50 years of waste containment, but the waste is expected to persist for millennia. So there's a disconnect there where I think it's important to know what's happening within those cells and within those spheres that are embedded in our freshwater environments. We're also looking at some mine tailings. We're looking at agricultural soils that are experiencing new dynamics over the winter season, with the freeze thaws happening more frequently because of climate change warming our winters. And the sort of common thread throughout all of it is to really understand how the microbes are responding to these anthropogenic changes to their environment, and whether or not there are any functions there that we can leverage or potentially inhibit in order to contain, transform, or better manage the waste that we're working with. And in the agricultural soil system, that's sort of managing fertilizer inputs so that we're making sure that winter fertilizer, with fall amended fertilizer, isn't consumed over the winter now that it's a warmer, more active season. And so we use a combination of multi-omics. We do metagenomics, metaproteomics, we have a little bit of metametabolomics. That field is maybe not quite ready for what we want to do with it. And then some cultivation-based work, so enrichment cultures, to prove activities or capture some novel organisms in the lab. And so it's an interesting mix. We do a little bit of field work, and then we have months and months of work on the computer in the lab following it. And one of the things that I really love about it is because we're not that focused on cultivating specific organisms, because we are often working with just the environmental sample itself, we're working with a little bit less bias as to what we then can observe within that system. And so that Tree of Life paper that you mentioned was really the result of many different groups working in different environments and identifying new radiations and placing each of those radiations onto the existing Tree of Life. And we wanted to say, okay, well then how do those all relate to each other? We need them all in one place so that we can see it. So it was really a project born of necessity. And one of the things I really love about it is that it was out of date. I think about 6 days after it was published, there was a new phylum within the archaea that wasn't represented on that tree. And in fact, the archaea have exploded in the meantime. So that tree is due for an update. Maybe don't tell the 3 people who have tattooed it onto their bodies. And also, if you were thinking of doing that, please don't do that. But it is this really interesting frontier right now. We're exploring what microbial diversity looks like and what microbial metabolic capacity looks like, and whether or not any of that are things that we can leverage to help start to clean up some of the mess that we're making.
Yeah, and one of the reasons, that just that paper and that particular figure you have in there, the new Tree of Life, is fascinating. It fascinates me because just looking at it, you instantly realize how little we know of, right? And the power of new tools and techniques that are out there now. How much can we, it just seems like unlimited. We can, if you're asking the right question and you're approaching the question the right way, and so that's, it really does explode in your mind when you look at it. At least, this is how I feel. So it is truly fascinating. It really does tell us how little we know, how much we need to find out about what's going on, and how life sort of works on this planet. So
yeah.
So Dr. Hug, the next question I have is well, it's related to science, but it's much broader question. I was wondering if you could give us some thoughts on state of equity, diversity, and inclusion, or EDI, and Canadian science, specifically Canadian microbiology.
I think it's a really interesting time for equity, diversity, and inclusion in Canadian science and in Canada in general. I think it's a moment with a lot of friction, and I think that is important. I think it's important with EDI issues that there is this discomfort or this friction around these changes, right? That this is a required component of grants now, but not just lip service anymore. It has to be something that has an executable plan. It has to have something where you need to be showing that you are committed to these ideals that you are espousing. And I think that requirement and that expectation growing is excellent. for science. There is strong empirical research that a more diverse team with different viewpoints and experiences will do better, stronger, and more productive work. It will be more replicable. There are great studies showing that a more inclusive and safe environment for everyone also leads to stronger teams, better work, better science. And I think that we should, in general, be striving for excellence. And one of the ways that we know we can achieve excellence is by ensuring that the great ideas and the strong scientists and the brains that we want to keep in Canada are safe in the spaces where they are working. And I think that part of that comes with a reckoning that that was not always true, and that is still not often true. And that is uncomfortable for those of us who have felt comfortable in these spaces or who have thrived under the existing systems, right? And so I think that every kind of change comes with discomfort. I think a lot of this is to look inward to understand where privilege has impacted success and to try to smooth that out such that, the great idea is going to move forward. The person with that motivation is going to be able to move forward. And that is acknowledging excellence and ensuring excellence, and that the metrics for that, I think a lot of science has often revolved around the model, the really strong model of the sort of lone wolf genius with probably a partner at home handling a lot of the general work, and likely a lab manager or administrative assistant or both at work handling a lot of the day-to-day administration and sort of trivial tasks. And that, that leaves the brain time to just think big thoughts and be very impactful in science. And I think that model isn't really the lived reality of most Canadian scientists at this time. It isn't really a functional model. It doesn't fit with how our society or our universities are set up right now. And so the sort of myth of the scientist and the reality of the scientist and the way that their teams and collaborative science and interdisciplinary science are building has this opportunity for us to look at the new system and say, how can we, as we rearrange our understanding of what a really successful scientist looks like to match what is actually happening, how can we also make space for that to be more equitable, to allow for, diversity to be comfortable, to not be rife with microaggressions, to be a safer space. And I think that reckoning is happening. And I think that I'm excited for it. I am active in EDI work at the University of Waterloo. And I think that it's good, but it's hard. It's hard sometimes. It's hard to look at your life and realize where things were easier for you than they were for others. It's hard to see things easier for others than they are for you because of societal systems and not because of aptitude or resources or ability or motivation. And so I think that it's an important conversation. I think it's going to be a long sort of arc, right? We're making progress. I think we're becoming more active in our progress as opposed to sort of acknowledging we're making changes. And I think those changes are going to take time to really build, but I'm excited for what it will mean for Canadian science.
Yeah, no, it is, as you said, it is actually quite exciting time, because the conversation that we are starting to have around EDI and EDI issues, if the conversation moves in the right direction, it will definitely lead to excellence. When I was a grad student 20 years ago, I had never heard of these terms because these were never discussed. So hopefully, if we have students graduating from our lab and our classrooms with knowledge on these issues, I think that's the very first step, education and sharing. And as you said, this is also just an issue of fairness, right? So nobody is talking about compromising excellence. The excellence is maintained where it is or gets better, but we make sure that every single excellent candidate or person is considered when we are looking for excellence, rather than just a subset of people. Yeah. So I agree. I completely agree. this is exciting time. I think the conversation that is sort of picking up. And I also commend some of the granting agencies that are actually requiring now for us to consider media, and not just lip service, not just writing a single line on your grant, but actually showing what it entails. So definitely exciting time. So, Dr. Hug, continue with our discussion on EDI. We do notice. So, for example, in our department now, a lot of younger scientists, younger trainees, now have families. This is sort of it's an interesting scenario where people maybe have doubts in terms of whether you can have both a career and a family together, or do you wait for one and then then go for other. I'll be honest, when I was actually a new faculty member, I was told by a senior person in my faculty that wait till you get your tenure before you decide to have a kid. So I cannot imagine female faculty members who what sort of things they hear. So any thoughts on that?
Yeah, lots. I mean, I'll preface by saying that I have two kids. I have a 5-year-old and a 2.5-year-old. And I had my first kid exactly to the day, 1 year after I started my tenure track position. But I had intended to have kids earlier, and it didn't work out on that timeline. So there's an aspect to this that can't really be planned. And I think that is a part that the biologists should better maybe understand. But I will say that I had mentors who were women and who had families through their careers, and that they had encouraged me to understand that having a family was something that was going to be a massive change in the way that you approach your life, in the way that you approach work, in the way that you approach the time that you have, and the priorities of how you're spending it, no matter when in your career you have those children, right? If it's during grad school, many women, many people have children in grad school. They complete their degrees, they go on to successful careers kind of thing, but it might, it will delay everything a little bit in terms of the time that it takes. But a kid is like such a bomb going off in your life that you can't really prepare for it. And there isn't a better or worse time to do it. I will say that the average age of achieving tenure, because it used to, I think also for women, be, make sure you've got your tenure before you have babies. The average age of tenure is in the 40s now. And that, again, harkens back to this earlier model being still what's in our psyche. And tenure was much more common in early 30s. Even a few decades ago. And now everything is pushed later, right? They're longer postdocs, graduate degrees are longer, pushing into that, 6-7 years instead of 4, 4-year postdocs instead of 2, starting a tenure track position in your late 30s. This isn't something that matches to a biological clock. And so I think your decision needs to be, do you want a family? And are you in a safe space to have a family? And is that, something that you want to prioritize? And if the answers to those are yes, it doesn't really matter what's happening outside of that for work, right? And it's going to change your approach to things. For me, it has delayed some of the work that I wanted to do. It has been a challenge to balance, but it has also made me ruthlessly efficient. And I am a far more effective scientist, and a scientist with a much stronger moral imperative than I was before I had kids. And I am more effective with the time that I have, and better able to balance competing demands on my time than I was before I had kids. And I have a stronger drive to look at, reducing methane emissions, handling plastic pollution, because I want to be able to look my kids in the eye when they realize what's happening and what's coming for them in the future, and say, I'm trying to help, I'm trying to work on this. And so for me, it has renewed my motivation in science, and it has improved my productivity. But there was a pretty big dip before that happened. Right. And it was worth it for me. It was something that I wanted. Yeah. And that isn't going to be true for everyone, and not everyone is going to be in the same space. But certainly in Canada, the social supports for new parents are pretty strong, and the universities are coming around to realizing that graduate students are a group that needs some of those supports directly inbuilt to their degree funding and things like that too. So it is an easier time to have a baby younger than it was, or earlier in your career than it was before. And it is also maybe more important to have them earlier because of these extending, stretching timelines. And so really, there is no right moment to have a baby, and it's not always right to have one. But if you do want one, then there's no reason not to. Certainly in science, there's no reason not to.
Yeah, and that's really well said. And actually, what you talked about, sort of our perspective on the science that we do, I concur. My own research on antibiotic resistance and the idea of antibiotics failing, and now I almost always think about, well, are we working enough to make sure that when our kids are a few years down the line, they have antibiotics for treatment. And the other thing that other very important thing that sort of I picked on is you said was safe space. And that is so important. I think it's all of us are responsible to ensure that there is safe space. If somebody wants to have a family, then they feel that yes, it's just not going to hurt their career. I mean, as you said, there are challenges, but it, the effect on career shouldn't be there. So
yeah. Exactly.
So this is great, Dr. Hug. Congratulations again on your award, and I really look forward to your talk at Guelph. This is going to be our first in-person meeting after a few years, so we are all looking forward to it, and we are all looking forward to seeing each other, and certainly your talk, maybe a new branch on the tree of life.
Maybe a couple more fleshed out. Yeah. So no, I'm really looking forward to it. I'm excited to see people again, and to have the opportunity to share some of the work that my group's been doing over the past few years.
Great. Thank you very much.
Thank you.